Hello everyone, my name is Lukas Voortman and I work in the Johnston Lab at Johns Hopkins University. I appreciate the opportunity to share with you my work identifying a two-step mechanism controlling stochastic sulfate specification in the fly eye. In the Drosophila visual system, differentiating R7 photoreceptors make a binary stochastic decision between two subtypes, characterized by their expression of either rhodopsin 3 or rhodopsin 4, shown here in blue and red. This decision is controlled by the stochastic expression of the transcription factor spineless. Each R7 precursor has a 67% chance to adopt the rhodopsin 4 fate, resulting in a consistent ratio of blue and red R7s, but a random pattern between eyes. While the outcome of this stochastic decision is observed in the adult eye, the choice occurs during the larval stage of fly development. Here's a schematic representation of the larval eye antennal imaginal disc. In the antenna, spineless is broadly expressed, making it a good internal control for our experiments. The eye has a more complex pattern of spineless expression. This tissue experiences a wave of differentiation from anterior to posterior, allowing us to observe multiple stages of eye development in the same tissue. At the crest of this wave, R7 precursors begin to differentiate and experience a pulse of spineless expression. Here, RNA is transcribed but not translated. Following this pulse of expression, these cells cease expressing spineless and undergo terminal differentiation. Once the mature state is reached, each cell makes an independent decision to remain repressed or reinitiate spineless expression, locking in that R7 subtype for the lifetime of the animal. We identified two enhancers termed early and late, which drive spineless expression in R7 precursors and mature R7s respectively, shown here using enhancer reporter constructs. To further characterize how these enhancers control the temporal expression of spineless, we use CRISPR to remove them and RNA fish to visualize spineless expression. As shown in the schematic, wild-type flies have broad expression in the antenna, a pulse of expression in R7 precursors, and 67% spineless expressing mature R7s. Interestingly, loss of early expression due to deletion of the early enhancer also results in the loss of expression late. Therefore, expression early is necessary for expression late. Moreover, we have identified cis and trans regulators that allow us to tune the level of spineless expression specifically in R7 precursors. In these lines, we see decreased expression early and a resultant decrease in spineless expressing mature R7s. Because the early and late enhancers drive expression in all cells at their specific state of differentiation, but only 67% express at the mature stage, repression must also play a role in spineless regulation. We identified two chromatin-modifying proteins that play a role in spineless activation. We therefore hypothesized that chromatin compaction may be playing a role in spineless repression. To visualize spineless compaction, I developed a three-color DNA fish assay which labels the spineless locus as well as the upstream and downstream regions with different fluorescently tagged oligos. Compaction is measured as the total distance between these three regions. With this assay, I found that spineless relaxes into a less compact state in cells where spineless is expressed. Interestingly, following early expression, the locus compacts when expression ceases, but does not revert to a fully compact state. This intermediate level of compaction is maintained to the mature state, which contains a mixture of spineless expressing and repressed R7s. Determining the compaction in those two subpopulations is an ongoing endeavor of this project. In order to identify how the temporal expression of spineless affects the compaction of the locus, I used this assay in the promoter and enhancer deletion flies. I found that in the absence of the promoter, the locus never relaxes from a compact state, suggesting that the transcription is required for spineless decompaction. Additionally, ablation of early expression causes the locus to remain compact in the eye beyond R7 precursors. These data suggest that the relaxing of the spineless locus due to early expression permits activation of spineless at the mature state by the late enhancer. In an endeavor to visualize spineless repression across the locus, I developed a repression reporter assay. Here, a reporter driven by a general eye enhancer has been inserted into different locations across the spineless locus. We hypothesized that repression of spineless would be sufficient to block expression of the reporter. Indeed, we find that in our control, the reporter is expressed in all photoreceptors. However, when the reporter is inserted into the spineless locus, expression is restricted to spineless expressing R7 photoreceptors. These data suggest that spineless repression affects the entire locus. Together, our findings support a two-step mechanism controlling the stochastic specification of the R7 photoreceptor. In step 1, a pulse of expression opens the gene locus. In step 2, repression and compaction limit spineless expression to a subset of R7 cells. These data highlight a dynamic, codependent regulation between expression and compaction in stochastic sulfate specification. I would like to conclude by thanking STB for the opportunity to present my work, the labs of Dr. Thomas Greger at Princeton and Dr. Andrew Gordis at Johns Hopkins for technical and image analysis support, the members of the Johnston Lab for feedback, and my sources of funding. Thank you for your time and attention.